Welcome to Click for IB Biology. We're studying topic B3.2, transport, and we're looking at transport in plants, and in particular, we're looking at the transport of water, the process known as transpiration. In this video, we're going to be concentrating on transpiration as it occurs in the leaf. There's a PDF of this presentation that's linked into the description below, where you can download that, annotate with your own notes, and then there's also 10 questions linked down there, 10 questions with answers, and you can use those to chart your own progress. Transpiration in the leaf. And so the image to the left is in fact showing the entire transpiration stream, with water entering at the roots, progressing up the stem, out along the petiole, to the leaf, where it evaporates through the stomatal pores into the surrounding atmosphere. Here we begin with the leaf itself. We're going to do it in the reverse order from that, as it's the leaf which is actually driving the transpiration stream. And this begins with the large surface area of the leaf absorbing light energy. And this light energy, in addition to powering photosynthesis that we would associate with the palisade mesophyll layer, this light energy, of course, as it's absorbed by the leaf tissues, is transformed into thermal energy. And that thermal energy is responsible for the phase change of water from its liquid phase into its vapor phase. This occurs down in the spongy mesophyll tissue, so that the water which is in this part of the leaf enters the gas phase, and this water vapor fills the space underneath the stomatal pores, which is often referred to as the substomatal airspace. And this substomatal airspace becomes near 100% saturated with water, with a water potential of approximately 0 kilopascals. The surrounding air, depending upon factors such as the humidity, will have a much lower water potential than is found in the substomatal airspace. And so what we therefore have is that for a distance thickness of one lower epidermal cell, we have an area of very high water potential and a area of very low water potential. And so we have a very steep gradient here. And so number 3 indicates the diffusion of water from the substomatal airspace into the surrounding atmosphere. As this process occurs, and because of the cohesion between the water molecules, water which is lost from the leaf is then replaced in the spongy mesophyll with water coming from the xylem in the veins of the leaf. The water molecules are cohering together through hydrogen bonding, and so as the water molecules are lost through the pore, this pulls the water molecules from the xylem into and around the spongy mesophyll, and so it is that water is lost from the leaf. The structure through which the water is lost are called stomata. And here we have an image of the lower epidermis showing a large number of stomata. And in fact, one of the calculations and exercises for IB biology is to actually calculate stomatal densities from real leaf tissues, and you'll probably perform that with your teacher in your laboratory. And stomatal density becomes an important variable in the actual rate of water loss from a leaf and from the plant. The individual stomata themselves are composed of 2 guard cells, which I'll outline the first one here, like so. And they are their cell walls are asymmetrically thickened, and on the inner wall of the pore, that wall, that cellulose wall, actually has extra thickening. The second guard cell is here, and so the actual stomatal pore itself lies between the two guard cells, and this pore can be opened and closed. And in general terms, we would think of the pore as being open during the day and closed at night. There are exceptions, and those will be studied at other points in the course. So we will progress with the idea of the stomatal pore being open during the day. And with the stomatal pore open during the day, in the substomatal air spaces labeled here, we have a near 100% saturation, and then in the surrounding air, as previously mentioned, we will have a much lower saturation depending upon the humidity of the atmosphere. And so we have a gradient over just one cell thickness. But interestingly, the diffusion from the pore, the most rapid diffusion, actually occurs at the edges of the pore. And so again, if we look at the pore from beneath, the circumference of the pore becomes an important feature in the actual rate of diffusion and loss of water from the leaf. And so it is that although we have a short diffusion distance with our lower epidermis here, and we have also determined that there's a steep diffusion gradient, the stomatal density has an important consequence, particularly if we include when that the actual size of the stomatal pores itself. And so if you have if we have small pores, that would give us a large density of stomata. This would give us a very large total circumference of stomatal pore, and that would give us a rapid rate of diffusion. So ideally, we would have a high density, so that means many stomata, and they would be small, and that would give us a particularly high total circumference from which rapid diffusion of the water vapor will occur. In the next video, we'll move on to consider this loss of water from the leaf has an impact on the stem, the movement of water in the stem of a plant. So that's the next video linked in on the left. Chance to subscribe to the channel if we'd greatly appreciate that. And on the right hand side is the playlist for your convenience.